Hello everyone. My name is Zhongyan Yu. I will present about extracellular electron transfer via microbial nanowires. Some microorganisms have a unique ability to transfer electrons to electron acceptors located outside their cellular boundaries. This process is not only fascinating but also crucial for maintaining the balance of various ecological systems. These microorganisms play a pivotal role in decomposing organic matter, breaking it down into simpler compounds, and facilitating the cycling of essential nutrients such as carbon, nitrogen, and sulfur in soil and sediment. One of the most significant electron acceptors utilized in these processes is iron oxide, a naturally occurring mineral that serves as a terminal electron acceptor in many anaerobic environments. Early research on electron transfer mechanisms to iron oxide primarily focused on a specific outer membrane protein known as a C-type cytochrome. This protein was thought to be critical for enabling the direct transfer of electrons from microbial cells to the iron oxide surface. Through this process, the mineral is reduced, playing an integral part in nutrient and energy flow within ecosystems. However, more recent studies have revealed surprising exceptions. Some microorganisms capable of reducing iron oxide lack this C-type cytochrome entirely. This discovery has prompted scientists to investigate alternative pathways and mechanisms that these organisms might employ to achieve electron transfer. The absence of C-type cytochromes in certain species challenges the traditional understanding of microbial respiration and highlights the incredible diversity and adaptability of microbial life. In various environments, microorganisms from the genus Geobacter have emerged as one of the most prominent and well-studied groups involved in iron reduction. These microbes are considered major contributors to the process of iron oxide reduction in natural settings. One fascinating aspect of Geobacter species is their reliance on direct contact with iron oxide surfaces to facilitate the electron transfer required for its reduction. Unlike microorganisms that use soluble electron shuttles or rely on diffusible compounds, Geobacter must physically interact with the iron oxide, making surface contact a critical step in their respiratory process. To enable this interaction, Geobacter produces specialized cellular structures known as PilA. These PilA serve multiple purposes but are particularly noteworthy for their role in bridging the physical gap between the microbial cell and the iron oxide surface. Acting as biological nanowires, these PilA allow for efficient electron transfer over distances that would otherwise hinder the reduction process. Furthermore, PilA production is not merely a structural adaptation but also a finely tuned biological response to environmental conditions. In iron-limited environments, Geobacter species can upregulate PilA synthesis to enhance their ability to access and reduce iron oxides effectively. Geobacter sulfurreducens exhibits a fascinating ability to produce PilA under specific environmental conditions, particularly in the presence of insoluble iron oxides. These PilA play a crucial role in facilitating direct electron transfer to the iron oxide surface. However, when soluble forms of iron such as iron citrate, are provided as electron acceptors, the production of PilA is notably absent. In these conditions, the cell appears to concentrate its electron transfer machinery on one side, which is attributed to the presence of a C-type cytochrome localized in the outer membrane. This suggests that the physiological response of sulfurreducens to different types of electron acceptors significantly influences its structural adaptations, such as PilA formation. Interestingly, the production of PilA in sulfurreducens can also be triggered under alternative environmental conditions. For instance, when fumarate is used as an alternative electron acceptor, the bacteria can still induce PilA formation if grown at suboptimal temperatures, such as 25 degrees Celsius, which is below its optimum growth temperature. This indicates that PilA production in G, sulfurreducens, is not only dependent on the type of electron acceptor but is also influenced by environmental factors like temperature. These observations suggest that the regulation of PilA production in G, sulfurreducens, might share similarities with other bacterial species where environmental and physiological cues control the expression of structural and functional components. The ability to modulate PilA production based on growth conditions and external stimuli demonstrates the adaptability. The genome sequence of Geobacter sulfurreducens contains two noteworthy open reading frames, ORFs, designated as GSU 1496 and GSU 1776. These ORFs are predicted to encode PilA proteins, which exhibit the characteristic amino acid sequences associated with type 4 PilA, a group of proteins essential for various structural and functional roles in bacteria. Type 4 PilAs are known for their involvement in motility, adhesion, and electron transfer, which are critical for the physiological processes of G, sulfurreducens. The presence of these ORFs highlights the organism's genetic capacity to produce specialized PilA proteins that contribute to its environmental adaptability and electron transfer capabilities. Detailed investigations into the functions of these ORFs have revealed significant distinctions between the proteins they encode. The protein produced by GSU 1776 shares similarities with PilAs associated with the type 2 secretion system, a molecular apparatus responsible for transporting proteins across the bacterial outer membrane. This protein, named OXPG, has been found to play an essential role in the secretion of proteins from the cytoplasm to the outer membrane, facilitating processes that are crucial for the cell's interaction with its external environment. This finding shows that OXPG specializes in the secretion of outer membrane proteins rather than the electron transfer function of PilA, suggesting that the role of GSU 1776 is independent of PilA. In contrast, the protein encoded by GSU 1496 exhibits features that align with PilA proteins found in other members of the Geobacteraceae family, such as Geobacter metalloreducens and Pelobacter prochionicus. Together, these PilA proteins form an independent evolutionary lineage distinct from those of other bacterial families. The PilAs produced by Geobacter species are notably shorter in length compared to those of other bacteria. Despite their reduced size, these PilAs possess a highly conserved N-terminal domain, which plays a pivotal role in PilA formation and function. This domain's conservation highlights its critical role in maintaining the structural integrity and functional efficiency of the PilAs. Additionally, the PilA proteins of Geobacter display lower overall conservation compared to those of other bacteria, reflecting unique structural and functional adaptations. For instance, while the PilAs of Shewanella wanidensis, a metal-reducing bacterium, exhibit distinct characteristics tailored for metal reduction, Geobacter PilAs are specialized for direct electron transfer processes. Notably, the genes required for PilA formation and assembly in Geobacter are located in close proximity to the PilA gene, a key genetic component involved in PilA synthesis. This clustered gene organization is similar to that observed in other bacteria, where related genes are often co-located to facilitate efficient coordination of protein production and assembly. The analysis of GSU 1496 revealed that it produces a PilA subunit protein named PilA, which plays a central role in PilA formation and assembly. This discovery not only provides insight into the molecular machinery of Geobacter sulfurreducens, but also underscores the evolutionary adaptations that enable this microorganism to thrive in environments requiring efficient electron transfer. All G. sulfurreducens strains were isogenic with the wild-type strain PCA, ATCC 51573. A PilA mutant strain was generated by replacement of the +61 to +159 coding region of the PilA gene, GSU 1496, with a chloramphenicol cassette. The PilA mutation was complemented in trans by introducing plasmid PRG5, PilA. When the PilA gene, a critical component for PilA protein production, was deleted in Geobacter sulfurreducens, the organism lost its ability to produce PilA. This structural deficiency rendered the bacteria incapable of reducing insoluble electron acceptors such as iron, 3, oxide and manganese, 4, oxide, which require direct contact for electron transfer. PilA, acting as conductive nanowires, are essential for bridging the physical gap between the cell and these insoluble mineral surfaces, facilitating effective electron transfer. Without functional PilA, sulfurreducens was unable to sustain this process. Interestingly, despite the inability to reduce insoluble electron acceptors, the PilA mutants retain the ability to reduce soluble electron acceptors such as fumaric acid and iron, 3, citrate. This indicates that the pilin protein encoded by the PilA gene is specifically required for extracellular electron transfer to insoluble surfaces, while intracellular electron transfer processes to soluble acceptors remain unaffected by its absence. Moreover, the mutants displayed the ability to grow on iron, 3, oxide medium when supplemented with either nitrilotriacetate, a chelating agent that dissolves iron, 3, oxide into a soluble form, or AQDS, an electron shuttle that facilitates electron transfer to insoluble substrates. These findings suggest that in the absence of PilA, alternative pathways or external aids like chelators and shuttles can bypass the need for direct contact with insoluble electron acceptors, enabling electron transfer and growth. In a subsequent experiment, the PilA gene function was reintroduced into the mutants. When grown on a medium containing acetate and fumarate at a suboptimal temperature of 25 degrees Celsius to stimulate PilA formation, the mutants regained their ability to assemble functional PilA. Consequently, their capacity to reduce insoluble iron, 3, oxide was fully restored. This restoration confirms that the presence of functional PilA, facilitated by the PilA gene, is indispensable for the reduction of insoluble iron, 3, oxide. In many microorganisms, type 4 PilA play a crucial role in attachment to various surfaces, aiding in colonization and biofilm formation. To investigate whether Geobacter sulfurreducens could similarly attach to iron, 3, oxide surfaces, an experiment was conducted where cells previously grown with fumaric acid were inoculated into a medium containing attached iron, 3, oxide. Initial observations showed that mutants lacking the PilA gene, which encodes the structural protein for PilA, were able to attach to iron, 3, oxide surfaces in a manner similar to the wild type during the first 24 hours of incubation. This indicates that attachment to iron, 3, oxide in the early stages is not strictly dependent on the presence of PilA. However, differences between the wild type and PilA mutants became evident during the subsequent 24 hours. The wild type cells continued to grow on the iron, 3, oxide surface, resulting in an increase in biomass. In contrast, the PilA mutants, despite their initial attachment, failed to grow further and exhibited a decrease in biomass. This disparity underscores the critical role of PilA in supporting growth when sulfurreducens relies on iron, 3, oxide as an electron acceptor. While initial adhesion to the surface may be PilA independent, sustained interaction and electron transfer to iron, 3, oxide require functional PilA. Further experimentation provided additional insights. When iron, 3, oxide was introduced to cells grown with fumaric acid, it was observed that PilA had a stronger binding affinity to iron, 3, oxide compared to the cells themselves. This suggests that PilA are specifically adapted for robust interaction with iron, 3, oxide surfaces. The ability of PilA to bind directly to the surface not only facilitates attachment but also positions the cells optimally for effective electron transfer. These findings indicate that the role of PilA in sulfurreducens extends beyond mere surface adhesion. The direct interaction between PilA and iron, 3, oxide likely enhances the efficiency of extracellular electron transfer, a critical process for the reduction of insoluble electron acceptors. The dual functionality of PilA in attachment and electron transfer highlights their importance in the survival and metabolic processes of sulfurreducens in environments where insoluble electron acceptors are prevalent. To measure the electrical conductivity of sulfurreducens through PilA, the microorganism was grown with fumarate, and then the PilA and other proteins were separated from the cell's surface and fixed to the graphite surface. This was analyzed with an atomic force microscope, which is a device that checks current flow using a conductive tip. The analysis showed that the filaments showed a strong current response when a voltage was applied. Positive current was observed when a positive voltage was applied, and negative current was observed when a negative voltage was applied, indicating that the PilA was able to conduct electricity well. Proteins other than the PilA, on the other hand, did not conduct electricity, and if these proteins were covering the PilA would be isolated and no current would flow. In particular, the linear ohmic relationship between current and voltage was maintained even when the voltage was changed while scanning a specific area of the PilA. These results showed that the PilA of sulfurreducens are highly conductive. For comparison, similar experiments were conducted with other microorganisms, as 1-densis, and Pseudomonas aeruginosa PilA, which showed no conductivity. Conductive PilA, often referred to as biological nanowires, enable cells to transfer electrons over significant distances beyond their immediate cellular boundary. This capability offers a distinct advantage in natural environments, such as soils and sediments, where iron, oxides are not uniformly distributed. These minerals are often sparsely dispersed across the surfaces of clay particles or other substrates, creating spatial challenges for electron transfer. The conductive PilA allow bacteria to bridge these physical gaps, ensuring efficient reduction of iron, oxides even in heterogeneous environments. The structural configuration of PilA is particularly suited to this function. In gram-negative bacteria, such as Geobacter species, PilA are anchored to both the outer membrane and the periplasm. This arrangement allows PilA to interact directly with electron transport proteins located in these regions. These electron transport proteins, which may vary depending on the microbial species, facilitate the transfer of electrons from intracellular metabolic pathways to the PilA. The diversity in cytochrome types and compositions among different iron, producing microorganisms reflects this variability in electron transport mechanisms. By serving as a conduit, PilA effectively complete the electron transport pathways, linking the cell's metabolic processes to external electron acceptors such as iron, oxides. Beyond their role in transferring electrons to iron, oxides, PilA also participate in other electron transfer reactions that are vital for microbial survival and interaction. For instance, in Geobacter species, PilA are often intertwined, enabling direct electron transfer between adjacent cells. This intercellular electron transfer facilitates cooperative metabolic activities, such as the sharing of electron acceptors in microbial communities, thereby enhancing the survival and efficiency of the population in resource-limited environments. The unique properties of PilA as conductive nanowires have exciting implications beyond natural ecosystems. These biologically generated structures have significant potential in the field of nanoelectronic technology. For example, their ability to conduct electrons over long distances could be harnessed in the development of bio-based electronic devices. Additionally, the genetic and biochemical adaptability of PilA proteins offers opportunities for engineering PilA with tailored properties. By manipulating the structure, composition, or conductivity of PilA, it may be possible to create nanowires with specialized functionalities for use in diverse applications, such as biosensors, energy storage systems, and bioelectrochemical devices. These multifaceted roles of PilA underscore their importance in microbial ecology and their potential utility in advancing sustainable technologies. Understanding the mechanisms by which pili mediate electron transfer and their adaptability in different environments could pave the way for innovative applications in both environmental and technological domains. Thank you for listening.